vicinity?
It often happens. Some summers we end up getting hurricanes and some we don't. It's been a little bit stressful for me because we've been remodeling our kitchen. So. Okay, this is weird, I guess.
Oh, we probably had the feedback from the.
Yeah, that was the feedback from the YouTube live stream, but now it's back.
Oh, okay. I don't usually see that. I guess I get in later.
Yes,
well, I guess one good thing for trips is that the U.S. stopped this thing of having to take a test to get back.
Yeah.
Well, you know, that was a real problem for me in Rome. Because I was there for such a short time to find a place, had to have Maurizio Bernori find a place that would test me, and I had to go and I had to get back, and it was annoying. And then of course the good news was I wasn't positive. If I was positive, I think I'd be probably still in Rome, you know.
Yeah,
there's a meeting, I think, in the Weizmann Institute in I think September that Kobe Levy's organizing. That's I was kind of worrying about. Jose says that he won't travel abroad now, or wasn't going to travel abroad as long as you had to do that test. So that's good.
Yeah, no, within Europe, we've not needed the tests for quite a while now, so it's good that the US joined.
Yeah, I guess, yeah.
So you don't need a test when you go back to the US. Yeah. Not even then.
That's right.
Starting now. Starting
now.
Day.
Starting Sunday, right. Right. I flew back on Saturday and needed to do the whole rigmarole, but Sunday it just disappeared.
Well, it probably was a good idea at one time, and I think a crazier thing is letting the people go without masks on the planes, and that's because of our legal system.
Right. You mean politics is involved here, Peter?
Yeah. I guess it goes back at least to Aristotle.
You're going to blame Europe.
I'm glad that I hear you say politics is involved. I've really gotten annoyed with this, that politics has become plural. So politics are involved, I hear a lot now. And I also hear people say the physics of the problem are this,
right. Or
the
really kinetics.
hate.
The kinetics
drives
are
me crazy. Right. And I'm trying to think, what is a single mathematic?
Right.
You know?
Or single kinetic.
Yeah. I know what a single physic is. That's a laxative. But
a physic is, isn't that like a dram of something, or?
Yeah, it's a laxative. I mean, the word physics, I mean, is from a Greek root which would have been pronounced pushik, pushika, and it really is the same word as push. So it has to do with things that are pushed around. So one thing you could push is what you push with a laxative.
Oh, geez.
So I think the numbers have stabilized by now. So okay. I guess we start now. So welcome everybody to our webinar on protein folding and dynamics. And it gives me really a great pleasure to welcome our speaker today, Michele Vendruscolo from the University of Cambridge. Here's another member who wants to get in. Just give me a second. Of course, many of you will be familiar already with Michele's work on protein folding, protein misfolding, and protein aggregation. And so in a way, Michele doesn't need a very long introduction. I nevertheless would like to sort of give you a flavor of his oeuvre. Now, simulating rare and transient states in protein dynamics has always been challenging. due to the sampling problem in molecular dynamics simulations, and that has been even more challenging so 20 years ago. And so to overcome some of these problems, Michele already 15 years ago used and developed an integrative approach in which he constrained molecular dynamics simulations using experimental data, mainly from NMR relaxation data, which really gave him detailed information about transient states of proteins, protein ubiquitin at the time. The approach by now is known as dynamic ensemble refinement. And this has been a starting point for a large array of different integrative approaches in structural biology. If we think about the whole field of intrinsically disordered proteins and Bayesian reweighting approaches, or fitting experimental data of IDPs using coarse-grained simulations. And so Michele's seminal work has really been a driver and a starting point for that. In addition, Michele is very active in developing and editing programs to predict essential characteristics of protein folding and aggregation processes. More recently, he also tied up with experimentalists, particularly the Knowles group, where he develops methods to kinetically analyze and dissect protein aggregation reactions down to the level of individual intermediates that allows him now also to identify small molecule compounds that could interact with those intermediates and potentially give us drugs against protein misfolding diseases. Now, a few words about Michele's CV. Michele studied physics at the University of Trieste, where he also received his PhD in condensed matter physics. He then moved more south to Israel, to the Weizmann Institute, where he did his postdoc for 2 years with Eitan Doumani at the physics department. And at that time, he already started working on proteins, analyzing protein contact maps and checking the possibility of using them to understand protein folding reactions. Afterwards, he moved to the UK to work with Chris Dobson at the University of Oxford. And since 2001, he's at the University of Cambridge, where he moved up the ranks and where he's now professor in biophysics in the chemistry department. Now, given all his seminal contributions, of course, Michele received many accolades. He was an Embo Young investigator. He became member of the Royal Society of Chemistry, from which he also received the Soft Matter and Biophysical Award. He's director of the Center for Protein Misfolding and Diseases and founder and chief scientific officer of REN Therapeutics. And so today, Michele will tell us how we can use knowledge on kinetics of protein aggregation to identify drugs against protein misfolding diseases. And we are all very much looking forward to your talk, Michele. But before I hand over the stage to you, let me remind everybody to please keep your mics switched off. During the talk, if you have a question during the talk, please use the chat window, type in question, and I will call you in the Q&A session afterwards. And so my final announcement is that the next talk in this series will be in 4 weeks from now, on July 11th. And there we will be welcoming Roland Netz from the Freie University of Berlin. Okay, and with this, Michele, thank you very much for joining us today. It's a pleasure to have you, and we are very much looking forward to your talk. So let me stop the screen sharing here, and the stage is going to be yours.
Thank you, Hagen, for the very kind introduction. Where is it? Trying to share the screen, disappeared somehow. Nearly there. Okay, should be ready.
That looks good.
Okay. So no, thank you again. I mean, it's a great pleasure to have the chance of giving one of these webinars. And, you know, they're very useful to the community, and so, you know, be part of them is an honor. So today I wanted to tell you about our research about one type of protein intermediates, as you said, Hagen. These are not the intermediates for folding, these are the intermediates for aggregation, but I'll come back to that in terms of drug discovery. So this work we carried out at the Center for Misfolding Diseases, which we co-founded with Chris Dobson and Thomas Knowles about nearly 10 years ago now, with the mission of studying and understanding the fundamental origins of protein misfolding diseases, and if we manage to use this understanding to develop diagnostics and therapeutics tools, which are largely missing at the present day for this type of diseases. The Center for Misfolding Diseases since 2018 is hosted in the new Chemistry of Health laboratory, which is a new building next to the main Department of Chemistry, main building of the Department of Chemistry. And this was funded really by a joint venture between the UK government and private investors, including benefactors, with the idea of bringing together academia with industry to facilitate the translation process from fundamental research to clinical applications. For this reason, in addition to the Center for Misfolding Diseases, the Chemistry of Health Incubator laboratory also includes an incubator space on the ground floor, and the space is currently occupied by REN Therapeutics, which is a company that we co-founded again with Chris and Thomas, in addition with Sarah Linsen Lund and Johnny Hatchke and Sam Cohen. So REN is taking forward the type of approach that I'm going to describe today for using chemical kinetics for drug discovery for diseases that involve protein aggregation. Historically, we started looking at Alzheimer's disease. So I wanted to give you a flavor of the type of challenges that one meets in trying to do drug discovery for this condition. So if one looks at the type of cellular processes that are perturbed in the disease, one cannot help but be overwhelmed, at least initially, because there is such a complexity in this disease that really is highly confusing. So we know that there is mitochondrial stress, there is production of oxidative reactive species. These are highly damaging in the long term to the cell. This is certainly going on. At the same time, there are also problems with the protein degradation pathways. The autophagy lysosomal system is typically compromised and effectively fails to remove proteins efficiently. Equally, the ubiquitin proteasome pathway is also impaired. So there is a problem with the protein cycle, so to speak. But also there is a problem with the inflammation. So this all these injuries at the cellular level imply that there is a sort of chronic inflammation that sets in, and eventually this is also highly neurotoxic. So eventually, in the long term, this is responsible for synaptic impairment and neuronal death. But in addition to all these processes, there is also protein misfolding and aggregation. This is really the reason why we started looking at this problem, to try and understand how this process is related to the disease and indeed to all other cellular dysfunctions. And the evidence that links protein aggregation with Alzheimer's is really overwhelming. So starting from the very initial description of the disease by Dr. Alzheimer, we know that in the brain of affected individuals, there are deposits that we now know are formed largely by proteins. And so we know that the amyloid plaques, which are extracellular, are formed by a variety of proteins, but mostly the amyloid beta peptide. And inside the neurons, there are other deposits which are called neurofibrillary rectangles, which are formed primarily by another protein, which is called tau. So we know that this is the hallmark at the molecular level of the disease. We don't really know whether this is a cause or a consequence of the disease, but this is what we are trying to establish. And similarly, we now know that protein aggregation is not unique to Alzheimer's disease, but there are a variety of other diseases that are caused or at least associated with the formation of protein aggregates, over 50 at the moment and counting as we discover new diseases or new links between aggregation and disease. I just mentioned Parkinson's disease, ALS, and then systemic conditions like or non-neurological conditions like type 2 diabetes and many others. So we hope that this type of approach that I'm describing today will be transferable to these other conditions. So the first question that we ask is where do the aggregates come from? Because we know, at least from the textbooks, that the normal fate of proteins is to fold and to function from the folded state. So after biosynthesis in the ribosome, they follow folding pathways and they're trafficked to their location in the cell or extracellularly, like in the figure shown here. And so this is the way proteins carry out their function, but we know that sometimes the folding process fails and proteins instead end up in some misfolded state. This in principle is a problem, but in practice we have a very effective quality control system, which is called the protein homeostasis system, that recognizes and removes the misfolded aggregates. In this case, shown here is the removal by the ubiquitin proteasome pathway. There are other pathways in the protein homeostasis system. So, this process of removal of the mistakes is quite effective until at least the protein homeostasis system itself is fully functional. So, however, as we age, the quality control system becomes progressively impaired. And what happens at that stage is that some of the misfolded proteins escape the control and tend to start to aggregate. And they can form either various amorphous aggregates or menacingly, some ordered aggregates, which are called amyloid fibrils. And in particular, on pathway to the formation of these amyloid fibrils, there are intermediate stages, which are small oligomers made up of, say, about 10 or 20 protein molecules, typically, that can be highly cytotoxic. But again, the quality control system is effective in removing these aggregates, so we can still, survive well, even in the presence of protein aggregation. That is until the protein homeostasis system is further impaired, and then effectively the protein aggregate starts accumulating, as I mentioned in the case of amyloid plaques and tau tangles. And so, in addition to this solid-like amyloid state, in the last 10 years, another type of protein condensate has been described, and we now know that is an extremely important state in the cellular context, because according to this phenomenon, proteins can form dense liquids, and in turn this is used in the cell to the formation of so-called membraneless organelles, which carry out a variety of functions. So this is a sort of liquid-like counterpart to the solid-like amyloid state. So we know that in addition to the native state, proteins can populate two condensed phases, the amyloid state and the liquid-like state. And so this prompts really the question: is there a driving force for the formation of these condensed states? So they don't form, by violating physical laws. They form because it's quite natural, perhaps in this idea, for proteins to populate these states. And so what we did in order to test this idea in a collaboration with Ulrich Hartl at the Max Planck Institute in Munich is to take worms and to do proteomics. And so in this experiment, we measured simultaneously both the total concentration, total abundance of proteins in worm lysates, and this is on the y-axis in this plot on the left, and the soluble abundance, so what is not in an aggregated state. And this is on the x-axis. So if all the proteins are soluble, so if aggregates are not forming at all, then every measurement would fit on the diagonal exactly, because the soluble abundance would correspond exactly to the total abundance. But what we found instead is that in the large majority of cases, over 3/4 of cases, proteins exhibited in the worms under normal conditions a fraction of aggregates. So say on average, perhaps 10% of the protein molecules are in the aggregated state. And this is independently from the sequence and the function of the proteins involved. So the protein aggregation is a phenomenon that happens at the proteome level. And so this really, from the point of view of thermodynamics, means that at the concentration present in the cell, the native state is only thermodynamically metastable, and the real thermodynamic minimum is the amyloid state. The reason why all proteins don't go directly into the amyloid state and remain functional for a while in the native state is the presence of a free energy barrier that makes the transition quite slow. So this idea that the concentration is so high that the amyloid state is the thermodynamic minimum corresponds to the fact that proteins are supersaturated, so above their critical concentration in the cellular condition. So this is a very good insight for both explaining the fundamental process of misfolding and aggregation in the cell, but also from the point of view of drug discovery, because we have seen that the protein homeostasis system is quite capable of preventing the conversion from the native state to the amyloid state, or in removing the amyloid state when it forms. So but the process eventually is inevitable. So proteins will convert into the amyloid state. So what we can hope from the point of view of drug discovery is to help the protein homeostasis system to either prevent the transition to the amyloid state or to remove the aggregates. So with this in mind, we start our drug discovery journey. And in order to sort of be more precise as possible in defining our target, we don't just want to target protein aggregation per se, but we want to target the oligomeric intermediates formed during the process. And this is because, as I said, there is evidence that these are the most toxic species. So we want to intervene on the process of aggregation in a way that minimizes the number of oligomeric intermediates that are formed. So the next question, because we have this target in mind, is we ask, where are the oligomers coming from? And it turns out that there are several ways in which oligomers can form. And these are, summarized in this scheme, at least some of the processes. So the first one is primary nucleation, and this is shown on the left side of this scheme. So you have protein molecules that come as monomers that come together to form oligomers. This process is called primary nucleation. This process tends to be quite slow, at least at the concentration that are present in the cell. And in fact, most of these oligomers can dissociate quite readily as well. If they don't dissociate, however, they can convert into some structures which are quite ordered. In fact, they have the typical cross-beta structure of the amyloid fibrils. So this step is called conversion. This is also quite slow. But if it happens, then the fibril seeds are formed, and then through another process, which is called elongation, monomers can add to the fibril ends and form longer and longer filaments or amyloid fibrils. And now, with the fibrils present, there is a striking additional phenomenon, which is the formation of additional oligomers on the surface of the fibrils. So, in fact, in this scheme, the fibrils act as catalyst for the formation of new oligomers. And so now this secondary nucleation process is responsible for the exponential proliferation of the aggregates, because here you have a reaction in which the rate is proportional to the amount of products. So the more products you have, the greater is the rate of the process. And so this is clearly very detrimental if the oligomeric intermediates are toxic. So even in this fairly simplified scheme of what happens during aggregation, because there are further processes that I have not described, you realize that it's already incredibly difficult from the point of view of drug discovery to understand what can be done, because here the aggregating proteins play different roles. So they are the reactants, because this is where the process starts as monomers. They are the products, because this is where the process ends in the fibrils. But at the same time, they are also the intermediates, and they are also as oligomers and the catalyst as in the fibrils. So these multiple roles means that if we want to stop proteins for aggregates, we have to either target all the roles. So we have to target them as reactants or as products or as intermediates or as catalysts, or at least choose the role that is mostly responsible for the production of oligomers. So we have to understand very well the balance between these different reactions. So this is a highly nonlinear reaction network, and we have to have that under high level of control. And again, from the point of view of traditional drug discovery, we have a major problem because, typically drug discovery works by screening compounds that have a certain binding affinity towards their targets, and then the binding affinity is improved progressively in the development of the compound. But here we don't really know where to bind because, as I said, the aggregating proteins play different roles. So we don't know whether to bind oligomers or bind monomers or to bind fibrils and so on and so forth. So, we have to find another strategy which is not based on binding. And because the rate of the reactions plays such an important role, we thought that if we understand how the kinetic networks work, and we use the suitable rates of the reactions as drug optimization parameter, we may make some inroad in the process. So from now on, the drug discovery will be based on the optimization of compounds for their ability to reduce their reaction rates, namely the reaction rates that lead to the formation of the oligomers. So how does it work in practice? We want to find drugs that minimize the population of oligomers. But the big problem in this program is that it's very, very difficult to measure the population of oligomers. So here we have a strategy that implies that we want to involves reducing the population of oligomers, but we cannot measure that population. So we have to find a way around, and the way around is to measure something that we know how to measure, which is not the intermediates in the reaction, that is the oligomers, but the products of the reaction, the fibrils. Now, because the fibrils are stable, is fairly easy, or not straightforward, to measure their mass. And so this is the first step in our procedure. The second procedure is to use chemical kinetics to extract from the measurement of the products some estimate of the amounts of intermediates, that is the oligomers that are produced. And because both the first and the second step are high throughput, we can screen compounds and optimize them. And then at the end of the optimization process, we have a compound which is estimated to be very potent in reducing the oligomer population, and we can finally measure directly by experimental means the population of oligomers. Now I'll come to the measurement of the population of oligomers because this is a very difficult step and very time consuming, but can be done. So the first step, I'll go through the 3 steps quite quickly if possible. So obviously there are many ways that one can conceive to measure the fibril mass, the amount of products in the reaction. But one method that works quite effectively is to use certain fluorescent dyes, or rather compounds that become fluorescent upon binding the amyloid fibrils. So one runs the reaction in the presence of these dyes. And as the aggregation takes place, one starts picking up a fluorescent signal. So, you have time on the x-axis, you have the fluorescent intensity on the y-axis, and you see as initially don't have aggregates, and then as the aggregation takes place, you pick up the fluorescent signal. The problem with this assay is that is hardly reproducible. So here we've shown, schematically, a few repeats, and you see that the time course of the reaction is different in all cases, although the conditions are the same, and therefore the reaction the measurements should be also the same. So this tells you that you have some more to do in order to do high throughput screening, because now if you want to find compounds that impact, decrease the aggregation, you have to distinguish this from the noise. So in this endeavor, it has been fundamental for us to work with Sarah Linz in Lund, because she really spent several years optimizing the assay conditions, including, for example, protein production, and the type of materials that are used in the plate readers to measure the fluorescence, and so on and so forth. A long series of the optimization protocols that have led to now the availability of highly reproducible assay that enables us not only to pick up compounds that delay the aggregation process, but they also do that in a concentration-dependent manner. So here there's shown several curves in the presence of increasing concentration of these inhibitor compounds. And so this is important because we have compounds that affect the aggregation reaction. But in fact, to our surprise, we find out that delaying the aggregation reaction, so the formation of fibrils, is not at all sufficient to take forward the compound. So what we found, without going into much details, is that even if you have 3 compounds that all inhibit the aggregation reaction, they may do so with very different outcomes on the population of oligomers. So the first compound may indeed delay the process of amyloid formation and also delay the process of, or reduce the amount of oligomers in the process. But maybe the second compounds can leave the population of oligomers totally unperturbed. So this compound would be totally ineffective in drug discovery because it doesn't remove the toxic particles. And there are even situations in which the compound may increase the oligomer population while decreasing the amount of fibrils produced. For example, a compound that stops the oligomer conversion will generate more oligomeric intermediates. And so that would be a disaster in drug discovery, because this drug, despite the fact that reduces aggregation, increases the number of toxic intermediates. So we really need to have the effect. So while we measure the population of fibers, we need to have the effect on the population of oligomers under tight control. And this is something that we can achieve by chemical kinetics. So this scheme here is another representation of the same scheme that I've shown before, and this shows how the protein aggregation process takes place in terms of the microscopic steps, so primary nucleation, secondary nucleation, and so on and so forth. Now, in chemical kinetics, one writes formally the equations that describe the time evolution of the various species, the oligomers and the fibers and the monomers involved in the aggregation reaction. And so one can keep track of this time evolution. And if one experimentally can measure some of the species in the reaction, typically the amount of products and the amount of monomers, one can extrapolate the population of all the other species. So you see that in this way, from the measurement of the population of fibers, we can backtrack the information to estimating the population of oligomers. This involves solving the equation, but by good fortune, this is actually possible by analytical methods, and this is the work mostly started by Thomas Knowles that has really shown that these equations are amenable to analytical treatment. So with this, we can relate measurable parameters, or parameters that can be fitted from the experimental data, to the rate of formation of oligomers, R, in this equation. So you see we have this rate of formation of oligomers is time dependent because the reaction of the nature of the reaction, but can be expressed in terms of the population of monomers, small case m, and the population of fibers, capital case M, plus certain reaction rates that we can fit from the data. So the bottom line is that for a given compound, we can calculate the rate of formation of oligomers, or the decrease in the rate of formation of oligomers that follows the treatment with this compound. So now we can implement a systematic drug discovery strategy by using the rate of formation of oligomers as the optimization parameter. And so starting from a given molecule, we can find derivatives that are of increasing potency. So this is our strategy. Now, the last bit that we need to check is that if we find a drug candidate, we have to measure explicitly the effect on the oligomer population, because so far through chemical kinetics we just estimated it, so we don't have really experimental data that we now want to gather. So there are probably a few dozen methods for measuring oligomers. They are all extremely difficult to carry out, and so this is still an open problem. So today I want to go to one particular solution that we have found, which is to generate antibodies that are specific to the oligomer. So they are conformation specific. So if you have this antibody in a sample that contains monomers, fibers, and oligomers, this antibody will not recognize monomers and fibers, but just the oligomers. So this is very handy in an aggregation reaction, because you can take aliquots and measure how many oligomers you have. So you can follow the time course of the oligomer population directly by using a readout based on this oligomer-specific antibody. So in order to get there, we decided to use a computational strategy, because it turns out that the oligomers as such are very difficult antigens, because they are transient, so they are not stable for for raising antibodies by traditional methods, and they are also extremely heterogeneous conformation. So the computational strategy involves, I'm going to describe, is sequence-based. So here there is the sequence of the A-beta peptide, in this case of 42 residues, and we selected 5 epitopes shown by the green boxes along the sequence. So these epitopes are selected to scan the entire sequence of the amyloid beta peptide, with the idea that at least one of them will be exposed in the oligomers, but not in the fibers, and therefore the antibodies will not recognize the fibers because the oligomers is not solvent exposed. So in order to find these antibodies, we use a database approach. I said computational, but really the computation is to go through the protein data bank and find matching fragments to those exhibited in the epitopes. So when we, perhaps in the previous slide is clearer, so we have the epitope in red, and we have the antibody corresponding antibody paratope in green. And so because we know the sequence of the epitope in red, we can scan the protein data bank for matching fragments in beta sheets. So this is a rather simple idea, but quite powerful, because this gives us matching peptides that form cross beta hydrogen bonds with the target epitope. And this completely bypasses a major problem in computational design, which is the knowledge of the force fields. In this case, there are no force fields, and it's just we take what nature gives us. So with this, we can generate a panel of 5 antibodies to scan the sequence of the amyloid beta peptide, and then we can test them for their effect on the aggregation reaction. So the 5 antibodies are in the 5 rows in this figure on the left, and we see that all 5 of them inhibit the aggregation reaction. So binding A-beta monomers in those epitopes can slow down process, but in ways that are very different in terms of oligomer population. For example, by binding to the N-terminus, we find that what is mostly affected is primary nucleation, whereas binding to the C-terminus is a primary effect on secondary nucleation. So with this observation, we wanted to check whether we can make even better in terms of computational design, and we started designing derivatives of, like chemical derivatives of the antibodies by replacing side chains with the non-natural counterparts. And we found that this type of strategy is quite effective in making antibodies that are more potent. For example, by replacing one side chain, we found, as shown in this slide, that is possible to enhance the reduction in the primary nucleation rates of 5 orders of magnitude. So this is a very useful tool to have available to get antibodies of greatly enhanced potency. So with that in mind, we selected an epitope towards the C-terminus that was very likely to be, because of the effect on the aggregation rates, to be exposed in the oligomers, but not on the fibrils. And then for this epitope, we designed 6 other sequences to the design method that I mentioned. And then we tested these 6 antibodies for their ability to bind oligomers. And in this case, we used an ELISA method. So the antibodies are immobilized on the surface, and then antibodies are tested for the ability to bind them. And we find one, which is shown in red in the bottom right corner, that is particularly better than the others. So with this, we can go on and measure the time evolution of the aggregation process. The important panel here is on the bottom right. So we have the unperturbed oligomer population in blue and the perturb oligomer population in red by the small molecules that is shown in panel D. So these small molecules delays the aggregation reaction, so reduces the rate of formation of fibers, and also reduce the population of oligomers that are produced in that reaction. And we can also measure the not just the effect of this drug-like compound in worms, but we also, so we can, by using transgenic worms that express the beta peptide, we can show that the compound restores the mobility of the worms, but also we can show that it does that by reducing the population of oligomers produced in the worms by using this oligomer-specific antibody. So with this, I just would like to conclude by saying that the drug discovery strategy that I presented was developed for A-beta aggregation in Alzheimer's disease, but because the process of aggregation is expected to be similar for other proteins, it can be likely applied to a variety of other diseases, and indeed we are already doing that for alpha-synuclein in Parkinson, for IAPP and ALS, sorry, in type 2 diabetes, and TDP-43 in ALS. With this, I thank many collaborators, even many more that are shown in this slide, that have worked in the Center for Misfolding Diseases and worldwide to our network on this program and in many others which are related. And thank you all for your attention.
Well, thank you very much, Michele, for wonderful talk. So let me start with the first question. You said in the beginning 10% of the, maybe I got it wrong, 10% of the proteins in a cell are generally aggregated. Is this correct? Or did you say 3/4 of them? Because you concluded from that percentage that essentially the native state is not the thermodynamic, is basically a metastable state and not the thermodynamically most stable state. And sort of it would make sense if it is 3/4, but it doesn't make much sense when if it's 10%. And sort of wanted to understand this.
Yeah, no. So
better.
yes, it's a good question. Is so 3/4 is the fraction of proteins that we've seen form aggregates. So by proteomics, we measured, if I remember correctly, 5,000 or 6,000 different types of proteins, and 3/4 of them were detected to form aggregates. Now, for each one of them, the amount of aggregates was a small fraction of the total, so about 10%. But let's say that in a proteome, 3/4 of the proteins tend to form aggregates, and the amount of aggregates for each one is about 10%.
All right.
Is that making sense?
Yeah. That makes sense. That makes sense.
Actually, I can explain, if we have just 1 minute, why we think that's the case. In fact, we call this observation life on the edge of protein solubility. And the reason is that effectively the cellular concentrations are close to the critical limit for aggregation. And this is not by chance, by some evolutionary drift. So if you think that you need a certain abundance of a given protein in the cell for functional reason, you may think that this protein is very soluble and it has no problem of aggregation. But you also know that you can have random mutations, and if these mutations don't affect the function, they may affect the solubility. And in fact, many mutations, because of the way the chemistry of the amino acid works, tend to reduce the solubility on average. But there is no force against this drift until you hit the solubility limit. So mutations that reduce the solubility are effectively, and do nothing else, are effectively neutral from the point of view of protein evolution until there is something that
Raises
happens
the
that is
concentration.
detrimental to the cell, and that is the aggregation. So for every protein, despite no matter what's the cellular concentration, there will be a drift that brings them to the edge of solubility. And so we believe that this is a general phenomenon, although admittedly we have only shown it in the case of this C. elegans.
Okay, yeah, thank you. Next question is by Amir Bitran. Amir, you want to unmute yourself?
Yeah, sure. Thank you. A very interesting talk. My question is just about why is it that an antibody that is specific for oligomers will actually decrease the levels and the rate of oligomer production, since at least naively, I would think that something that binds to an oligomer would actually stabilize it, or maybe kind of sterically block subsequent aggregation and keep it in the oligomeric state. So why is it actually having that decreasing effect?
Yes, that's a great question. So this is, I mean, one of the shortcomings of using chemical kinetics, because we don't have a direct insight of, what these oligomers bind to and how they actually affect. So we don't have a mechanistic understanding. We only measure the effect on the reaction rates. So one can do further experiments to answer to your question that involve binding, but we haven't done them.
Thank you.
Next question by Ben.
Yeah, thank you very much, Michele, for this beautiful overview. I guess one of the obvious questions is, what are the hottest candidates in terms of toxicity of the oligomers? Which processes do you think are most likely to be affected by the presence of oligomers? What does make them toxic?
I mean, here, there is lots and lots of evidence from model systems. What happens in the brain is, almost totally unknown. So we have, in vitro evidence from either, cellular studies, for example, that indicates that oligomers, because of the exposed hydrophobic surfaces that can bind to lipid membranes and perturb their integrity in various ways. So they, calcium tend to, go through upon binding to the oligomers and so on. Or they can also bind cell receptors and trigger, other cellular processes. But really, if you ask me what happens in the brain, that's still unknown. And this is a major question mark on the entire approach, because we have to effectively. We have some evidence that, even good evidence perhaps, but it's still no direct proof that the oligomers are toxic in disease, and therefore we need to test the hypothesis by actually testing the effect on the cognition upon treatment of compounds that reduce the oligomers.
Mm-hmm. Thank you.
Next question is by David Nesbitt. David, please.
Thank you, Michele. That was really, opened my eyes. Certainly, the most amazing thing that I take away is what a large fraction of the proteins are right there at the solubility limit. And so my question is, one would presume that even sort of relatively simple issues such as crowding would have an important phenomenological impact on these sorts of solubility issues. Is there some way in which molecular crowding by other species has impact on this solubility property?
Yeah, absolutely. I mean, our understanding of this effect that you just mentioned is dramatically increased in the last few years when people have started studying the phenomenon of protein liquid-liquid phase separation, because then the control of this transition is a very direct functional relevance in the cell. And so we are starting to understand how the modulation of the phase boundary, including by effectively crowding agents, in the cell. They are not really crowding agent per se, but they are. You call them crowding agents if you are a biophysicist, but otherwise they have very specific names to biologists. Exactly, but anyway, right. So the point is that this modulation that you've mentioned is a very high importance in cell biology, and the phenomenon of amyloid aggregation can take place within the dense liquid aggregates, and so in turn these modulating agents also affect the downstream the process of amyloid aggregation. So absolutely.
Yeah. I apologize for being a biophysicist and just using a simple description, but that's very, very helpful. Thank you.
Okay, so the next question is by Julia Stegner. Julia, please.
Hi, yeah, I was wondering how difficult was it to design an antibody that didn't have any cross-reactivity with either the monomer or the fibril? Does it require the oligomer to have very different conformations from the fibrils themselves?
Yes, that's a great question. So because it's very difficult to carry out structural studies on the oligomers because they are transient and heterogeneous. We didn't know which epitopes are exposed in the oligomers, but not in the fibrils. So we did the sequence scanning. So so we found that this antibody that binds the oligomers, much better than the fibers, and therefore we can deduce that that particular epitope is not exposed on the fibers. Of course, that epitope will be exposed on the monomer, but because the monomer is disordered in the case of A beta, there is a very high entropic penalty to binding the antibody. So although the epitope is exposed both on the monomers and on the oligomers, in the oligomers, because the oligomers is structured, the entropy of the oligomer is much smaller than the entropy of the monomer, and therefore the binding can be tighter with the oligomer rather than the monomer. So the antibody is oligomer specific for different reasons, from fibrils to monomers. In the case of fibrils, the epitope is not exposed. In the case of monomers, is exposed, but there is a high entropic penalty.
Okay, thank you very much.
Next question is by Eugene Cheknovich. Eugene, please.
Hi, Michele. Nice to see you and beautiful talk. Thank you very much. My question is about this concept of life on the edge of solubility, and my understanding that you view it as a solubility against homotypic aggregation, but there could be a lot of variants of heterotypic aggregation or association between proteins, between different proteins, in particular leading to liquid-liquid phase separation. So I'm wondering why this is on the edge of homotypic solubility, but not on the edge of liquid-liquid phase separation, generally speaking, or maybe it's also on liquid-liquid phase separation. So how do you distinguish here between LLPS and homotypic aggregation in this?
Yes, hi Eugene, nice to see you too. That's a really great question. This idea, this hypothesis of life on the edge of solubility, if it is true, it is how can I put it, an average idea, in the sense that any specific system, any specific proteins will have its own rules, so to speak. So in particular, so there are actually many points to your question. Let me address first the main one about what is the liquid condensation has to do with the solid condensation. I think because the concentration of proteins in the liquid phase and in the solid phase is similar, effectively you should be able to translate the life on the edge of solubility to the life on the edge of condensation. And then, of course, although this is the general idea, then every specific protein will have its own rules, as I said, and certain proteins we know that they don't spontaneously phase separate by themselves, but they really need partners. So in this case, this hypothesis will have to be modified to some extent. But I would say quite generally, it is always heterogeneous, the system in the cell. So when I say critical concentration, is always critical concentration in the cellular context. So, there are so many things to be understood that I share with you actually many of the questions. So when I say cellular context, obviously we know that there isn't one cellular context, and therefore the critical concentration must be hugely variable from cell to cell and cellular location to cellular location. So I think this idea, personally think that is likely to be true for the fundamental reason that I mentioned about protein evolution. But how exactly that works in every specific case, I'm not sure. Is this helpful, Eugene? I mean, I know I haven't.
Yeah, no, I think it's helpful. And I think we have also some evidence that could apply to heterogeneous, at least at some limited kind of level to heterotypic aggregation, because kind of you can look at this various combinations. So yeah, I think it's quite helpful. Thank you. Yeah.
Yes. I'm sure that it is right. In fact it has to hold for heterogeneous aggregation as well.
Okay, Ben has another question. Ben, please.
Yeah, if I may, I have sort of a technical question, which is regarding the assays that can be done to identify the role of drugs or whatever you add to reduce oligomer formation or whatever you want to reduce. I mean, it appears that if you look at A-beta aggregation, or probably that's true for most of these aggregation reactions, they're extremely sensitive to solution conditions, presence of surfaces, heaven knows what else. And so I was wondering, what are the best tricks that you can use to reduce this variability and identify robust drugs or robust patterns in aggregation kinetics?
So, but are you asking me if we can find compounds that work under many different conditions?
In some way, yes, or asked in a different way, if three people in different labs would do the same assay under slightly different experimental conditions, they might find different things, right? And so the question is, how robust are these assays, or how can you make them as robust as possible so that you know that the effect you're seeing is not just sort of one aspect that depends very much on the specific conditions you're using in this experiment?
Yes, I mean, can I separate your question into different ones? Because I think they're both relevant. So one is that the aspect of drug discovery, because we would like, because the protein aggregation process is so dependent on details, you don't want to drugs that work only in very specific conditions. So you want to carry out testing of the drugs under many different conditions, as many as possible. So you want not one assay, but many assays, and you want them all reproducible. And this is the second part of your question, how you make them reproducible. As I said, that this is, a huge amount of contribution from Sarah Lindsay, and there are many details, and these are published in various papers by Sarah or together with her from us. And but one absolutely essential one is to make the proteins in a recombinant expression system, because if you buy the proteins from vendors, they tend to contain impurities, and the impurities, because of the variability, the sensitivity to the conditions, obviously affect the process. And then another important parameter is whether you shake to induce aggregation. This is done quite frequently. But in what we found out that shaking tends to create liquid air-water interfaces, and interfaces themselves tend to act as catalysts for the aggregation process itself. So depending on the amount of air-water surfaces that you create by shaking, you have a faster or slower primary nucleation process, and so on and so forth.
Yeah, thanks very much, Michele.
And the next question is by Liz Meyering. Good to see you, Liz.
Hi, Halligan. It's a really interesting discussion. And I agree with all these things that have been discussed. I guess one question I have is, and I like the characterization with many antibodies and looking for reproducibility and so on. I guess a question I have is, do you think that there's anything sort of in the early phases that may be common with your in vivo, in vitro sort of systems that might help? Do you think that that's possible?
Early phases of the
very initial steps of aggregation, I guess I'm wondering, right? So you can have all kinds of things that happen. It can all take off on you. And I'm wondering, if you go earlier, is there anything that might be more targetable there? Like the very initial steps of self-association. Is there, that might be common, more often common in different systems? It's a big speculation. I know about, we all know about all the complexity, I guess, and we heard a lot about it just now too, but the complexity that can happen. And we know that aggregation is very sensitive to different conditions. And the question is, if and when are there ever any sort of common aspects that we could target more effectively?
So are you thinking of drugs, potential drugs that may work for different proteins that aggregate?
No, I was thinking for one particular protein. I guess you could think broader. I mean, hydrophobic things are important, but that's I think too general. Yeah, I guess I was thinking.
Although this is not what I discussed today, an alternative avenue that we are exploring for drug discovery is to stabilize the native state of proteins. So now, stabilizing native state of proteins is very traditional drug discovery, but not if the native proteins are intrinsically disordered, like A beta or tau or alpha-synuclein or APP, major players in different misfolding diseases. So stabilizing the native state of disordered proteins by small molecules seems to be possible. At least our initial results on A beta are indicating that. And so that's the type of general approach that you may be hinting at. Is at least one way.
I mean, I agree. I think that's a nice approach. No, I was actually thinking more generally in terms of, when do all the pathways diverge, and can you go earlier at them?
Yes, but
but that is it. The native state is sort of the simplest at the very beginning. Yeah.
No, but the very beginning is the native state. So you have to stabilize that, because if you bind to other states, you stabilize those other states. So you don't want to bind oligomers because the bound complex will be more stable than the isolated oligomers. And so if you intervene after the native state, after the monomeric state, I cannot see how you can be beneficial. So this is why I said my understanding of the approach that you are suggesting is stabilize the native state.
Yeah. Thank you.
So Michele, I don't see any further questions. So let me ask the final one, which is also a rather technical question. So people in the field of aggregation and fibril formation use THD fluorescence since, I don't know, 30 years or something like this. And I understand that it's a very effective way of monitoring fibril mass and fibril formation. But I was wondering, with all the techniques that came that were developed in the past 5 to 10 years, I'm thinking of mass photometry, for example, ISCAT. Wouldn't this be a good alternative technique to identify oligomeric species and count them directly in an aggregation process, or is this totally useless for aggregation, for monitoring, quantifying aggregation kinetics?
No, not at all. I mean, you're right. I mean, there are alternative methods that can give you a readout not just on the fibrils but also on the oligomeric intermediates, and that would be much better. The concept is spot on. The implementation is still, to my knowledge, a bit lagging behind because you need a scalable approach, a high throughput approach, and none of the methods, to my knowledge, are already at that stage. But certainly that's the direction to go.
Right. To get direct numbers of oligomers.
Yes, high throughput oligomer measurements is the holy grail in this particular drug discovery strategy. Yes.
Okay. Thank you very much, Michele. I don't see any further question. So thank you very much, Michele, for wonderful presentation and a good discussion. And thanks all of you for joining today. And we hope we see you again in 4 weeks from now with Roland Netz as our speaker. And till then, thanks a lot, Michele, for joining.
Thank you. Thank you all. Thank you again.
Thank you very much. Goodbye, everyone.
Goodbye, everyone.
Bye.